Hello dear students, I hope you have understood about the last class. Today we are continuing our studies on cell interaction. The topic which we are going to see today is cell dissociation. So let's see what is cell dissociation. Cell dissociation means separating of the cells so that we can get a single cell. And then why is it important is these cells can be used for the further research like in the field of biomedical or in the field of cell biology. And this also plays a very important role where it has a wide range of application in a tissue transplant and as well as a cell therapy. Once the cell is being dissociated, where we get a single cell, they are used for the primary cell culture and they are used for the secondary culture, and these cultures are used for the study work, that is for a biomedical research or for the cell biological studies. Here you can see a picture where you can see the cells are attached with each other, that is intracellularly and with the matrix also. So these are linked with the help of certain proteins. Breaking up of these protein, the cells are being released so that we can isolate a single cell. And coming on to the types of cell dissociation, there are three types: the enzymatic, chemical and mechanical method. In an enzymatic method, we use certain enzymes such as trypsin and chymotrypsin. There are certain chances where we use a combination of these two enzymes to break up the protein bond so that we can get a single cell. And next type is a chemical cell dissociation. In this type, we use two types of chemical, that is EDTA or EGTA. Why? Because these cells are linked with each other where linkage is involved or the linkage is carried out with the help of cations. These cations play a major role in linking of these two cells or linking of the cell and the matrix. So for all of these, the cations are involved. So the chemical like EDTA and EGTA, they go and bind to this cation and disrupt the bonding so that we can isolate a cell. Next, moving on to the mechanical dissociation. Here we take a tissue, we scrap it, cut it, scratch it into a smaller pieces and they are washed to separate the cells from the tissues. Sometimes they are subjected to gentle agitation so that we can isolate a single cell. After knowing a different kinds of cell dissociation method, here we'll study in detail about one of the cell dissociation method, that is a enzymatic method. So the enzyme which we study here is a trypsin, and the process where the cell dissociation process is carried out by using a trypsin is called as a trypsinization. And this is one of the proteolytic enzyme, which means it has the capacity to break the protein bonds or the linkage between the cells. So this is the 3D structure of the enzyme trypsin. So let's see what are the types of trypsinization. Here you can see a warm trypsinization. In warm trypsinization, what you do is you take a tissue, wash it with a dissection basil salt solution. After washing it with that, you transfer the tissue into a flask, and to that a warm trypsin, which is about 36 degrees centigrade, is added to that, and after addition of this warm trypsin you keep it undisturbed for 30 minutes. After that you can see a dissociated cells on the supernatant, which is the upper part of the solution. You collect it and again you have to add a fresh trypsin to the mixture, and this procedure is repeated for about every 3 to 4 hours. And then you can remove the trypsin from the tissue by the help of centrifugation. And coming on to the cold trypsinization method, in cold trypsinization method, you take the tissue, wash it with a dissection basil salt solution, and after washing it, you transfer the tissue into a flask. To that, a cold trypsin, which is about 4 degrees centigrade, is added, and then you soak it there for about 6 to 24 hours. After when soaking hours is completed, you incubate the tissue with the residual remaining trypsin at about 36.5 degree centigrade, that too for 20 to 30 minutes. And by repeated pipetting, when you do the pipetting again and again, you can see the dispersed dissociated cells, and the trypsin can be removed from the tissue by the help of centrifugation. So after the process of trypsinization, it is important to remove the trypsin from the cells so that it does not harm the cells. So till now we studied about the process of trypsinization, that is cell dissociation. In a further slides we'll be studying about the contact inhibition of the cells.